Hello. Well, I'm glad to be here, and thank you all for coming. I'm Larry+Nogee. I am a neonatologist, so I take care of newborns. I'm not a pulmonologist, nor am I a geneticist, although I sort of pretend to be, I guess, sometimes. But what Anne asked me to talk about, and what my colleague Aaron are going to talk about, is how we go about finding a causative gene for a child disorder and why that may be important. So what I'm going to give is a sort of a little primer on genetics and try and review some basics and some basic terminologies and then sort of the strategies of how we go about doing this. So one of the things to start off with is sort of an evolving picture of how we look at health and illness, and really looking at them at a continuum or a balance between wellness and illness as the combination of everybody's genetic predisposition and the specific environment that you're exposed to. And this is what we're actually teaching our medical students now. Not to think of illness as, why did something unlucky happen to this person, but what is it about a person's particular genetic makeup? What is it about their environment, so that the combination is that they sort of shift the balance from illness to wellness at this point in time. And there are some things that we think have more of a genetic contribution, and that's what I've shown here, so surfactant protein B and ABCA3. And then there are other things that we think of as not being genetic at all, such as I've got auto accidents at the far right, but in fact your hand-eye coordination, which is partly genetically determined, your risk-taking behavior, which is genetically determined, impacts on whether you're likely to have a car accident or not. So everything's genetic, everything's environmental. And that's a little bit of an oversimplification, but the point is that illness and wellness are going to depend on a very complicated interaction between the two. So why do we want to find genes? Well, it may provide an explanation for an illness when there wasn't previously one. It can help us understand how and why that disease occurs. It allows us to start to classify illnesses based upon what their actual causes are, as opposed to trying to sort of group symptoms or findings on a lung biopsy or findings on other laboratory tests, because we know that there are some illnesses that can manifest in very different ways. So the symptoms can be the same, but the underlying cause may be very different, and the approach may be very different. We'd also like to be non-invasive. So if we can do this on a little blood test or a little sample of saliva, as opposed to having to take a piece of lung tissue, that's something that we'd like to be able to do. And we'd like to be able to give you, the families, as much information as possible so that we can counsel you on what are the odds of other family members having problems, what are the odds if you have another child of the same problem occurring again. Can we get some information on what's likely to happen in the future? And then finally, we hope that if we have some understanding of the root cause of the illness, that's going to provide us a mechanism for starting to develop some new treatments. So let's talk about a few definitions, and I apologize if there are biologists in the audience, and this is oversimplification, but I can assure you that there are plenty of doctors and medical students who get confused about all this. So a gene is a basic inherited unit of information. It's essentially a blueprint or set of instructions for assembling a protein. Genes are made of DNA, and DNA is actually a fairly simple structure. It's 4 bases, A, C, G, or T, that are just strung together in a very long chain. And the DNA is grouped into chromosomes. Humans have 23 pairs of chromosomes. They vary in size. 2 of them determine whether you're a boy or a girl. But there are 3 billion individual pieces of DNA in the human genome. That is a big number, okay? And every one of us has some 20 to 30,000 genes. The number keeps changing a bit, of which only about a third we actually know what they're doing, okay? And the other important thing to realize is that everyone has some genes that don't do what they're supposed to do, okay? Some of us know what those genes are, Most of us don't. And fortunately, most of those don't cause any real health problems. They just, are there, or they may determine things like hair color or eye color that are individual differences, but that we don't think of as health or disease. So genes, I've already said, they're the section of DNA that tells the body how to make a protein. There's an intermediate there. The gene actually has to be copied over into a molecule that's called a messenger RNA, and that's what actually is translated into a protein. And the proteins are the basic building blocks and machineries for everything in the cells, and they're both the structure and they're the machinery, the functions for how the cells work. And then we talk about phenotype, which is the physical expression of what the sum of all this is. So DNA is a double helix. This is what Watson and Crick won the Nobel Prize for discovering the structure of DNA in the early 1950s. And each strand is actually a complete complement of one another. So you take that genetic code in one direction, and you know what the code is in the opposite direction because C always pairs with G and A always pairs with T. And this is a very important principle in biology because this is how genes are able to be copied. Because when they separate, each one can provide the information for making an exact copy of itself. And the order of these bases is really like a language. It's a code for what determines how to make a protein. And this genetic code is read in blocks of 3. Now, a large percentage of the DNA actually doesn't code for anything. In fact, only about 1 to 1.5% is actually considered the coding sequence. And what the rest of the 99% is, we don't really know. It's important in controlling genes. It used to be called junk DNA, and that term is still around. It's not a very good term because it's not junk. It has an very important function, but we don't know what the functions are. And that remains an important mystery. But an important part of that is that there are lots of changes between individuals that occur out in those regions, and we currently have very little understanding of what those changes mean. And then the entire genome has to be copied over all 3 billion bases every time you make a new cell in your body. Okay, so think about that. Your homework assignment tonight is to go home and to copy the entire encyclopedia letter for letter. And when you're done, is it going to be a perfect copy of the original? Well, cells aren't perfect. They make mistakes, and that's how things change. So what can go wrong? Well, all or part of a gene might be missing or duplicated. Okay, you got a little distracted, there was an exciting part of the hockey game going on, and you just forgot to copy chapter 3. Okay, or you wrote it down twice. Okay, a little piece may get turned around. So by mistake, you sort of flip a page and you write things backwards. A small piece may be missing or inserted. You added an extra letter here or there, you just left 1 or 2 out. Okay, there can be just a single base. One single letter is changed. So instead of writing fox, you wrote fix. And it sort of made sense when you looked at it, but proofreading it, it might change the meaning. Now, when those changes occur, the term is called a mutation. And mutation just means a change from the usual sequence. Colloquial mutation becomes associated with illness. Polymorphism is a change that by definition is found on more than 1% of the copies of a gene that are out there. And that's just a statistical definition. So it's something that differs from one person to the next, and more than 1% of the population has it. It's considered a polymorphism. But actually, each of these terms doesn't necessarily imply by itself anything to do with health or disease. And another important thing to understand is how genes are passed on and how they cause illness. There are different patterns. And there are 2 basic patterns. The first is called recessive. And in this case, there's usually no family history of disease. And the reason that is because these usually occur when one copy of the gene doesn't work correctly. And for most of the genes in our body, we have 2 copies, one we got from the mom, one we got from the dad. And as long as one of those copies is doing its job, you got a perfectly working protein that's doing what it's supposed to do, and it doesn't matter that the other one isn't working. So it's only when a child happens to get two copies of the gene, one from mom, one from dad, that each don't do what they're supposed to do, that these kind of illnesses show up. And in fact, you have a 50-50 chance of passing on each copy of any gene that you've got. So if you do the arithmetic, what that means is if each parent has one copy that doesn't do what it's supposed to, there's only a 1 in 4 chance that the child's going to get the combination where both don't work. So that's why usually for recessive diseases, there may not be any family history, or you may have a large number of children and only 1 or 2 have the illness. In contrast, a dominantly transmitted mutation or a dominant pattern usually affects multiple generations. So you may have grandparents, you may have an aunt or uncle or a cousin, you may have a sibling, but there are usually multiple people at each level of the family tree that have the illness. Usually in this case, only one copy of the gene needs to sort of malfunction in order to cause the illness. It can get confusing because there's something called incomplete or variable penetrance. What that means is that the gene itself isn't the sole determinant of whether there's going to be illness. Something else matters, either environment, viruses, smoking exposure, some other gene, so that two people can have, even within the same family, the exact same copy of the gene, the exact same problem, and may have very different physical consequences for their health. And then lastly, we need to separate out whether these are on what are called the autosomes, which are the first 22 sets of chromosomes, or on the sex chromosomes, or X-linked. If you have 2 X's, you're a girl. If you have an X and a Y, you're a boy, because that changes the likelihood of sort of causing illness and whether it's going to be in just boys or just girls. Why have I spent time on this? Because when we try and figure out, whether there's a genetic cause for a disease, if we don't understand these inheritance patterns, we can very easily get misled. So how do we go about and find genes for illness? Well, there are 2 basic strategies. The first is what's called a candidate gene approach. So we start with a gene of interest, and we try and figure out if it has a role in that illness. Okay, this is really an educated guess. All right, we take a gene and we say, because of what we think this gene does, and that may or may not be accurate, or where it's made in the body, or because somebody's made a mouse and they've inactivated that gene and that mouse gets something that looks like a child illness, that we think that this is a good candidate for a lung illness. So an example here is ABCA3. We looked at ABCA3 because we knew that gene was a cousin to the gene that's involved in cystic fibrosis. So it was involved in a lung illness. It's made in lung cells, and where it's made in lung cells is an important part of the cell for how surfactant material is processed. And we knew that other cousins of that gene are also involved in other genetic illnesses and involved in the kind of shuttling kinds of chemicals that are important in surfactant. So it seemed like a good candidate for explaining lung illness in children whose problems might be related to surfactant disorders, and that in fact turned out to be the case. The other broad mechanism where we look for genes is what's called linkage. And here we start with an illness, and then we try and find the causative gene by studying people and families, in which the illness occurs, and then trying to find out what tracks with the illness and what doesn't track with the illness. So the old way that this was done, we just had panels of differences in the genes, that are scattered throughout all 3 billion base pairs. And there are various ways of doing this, ranging from looking at different patterns on gels to chips, where you can actually look at 600,000 of these at a time. And you look through and see, well, if we've got a very large family, okay, who in the family has this change, who doesn't? And can we find something that sort of is going along with the illness, and does that get us close then to the gene that's responsible? Now, there are some caveats here. You need to understand how that gene is being transmitted, what the inheritance pattern is. Because if you assume it's recessive and it's dominant, then you can totally miss the boat. You need to be certain who truly does and doesn't have the illness. And that's where this problem of incomplete penetrance comes up, because you may assume somebody doesn't have the illness when in fact, and they may not, but they may have the same gene change, and so therefore you throw that gene out and then you've made a mistake. You often need very large families in order to be able to pull this off. And then to make things even more confusing, different genes can cause the exact same kind of illness. So you can think you have two families where you have the same illness and they have two different genetic causes. But then when you try and sort this out through this linkage approach, you're going to get very confused. So what do we do when we find a gene? What are some of the pros and cons of genetic testing? Well, it's an explanation for illness. Diagnostic testing becomes feasible, but interpretation isn't often straightforward. It's expensive. It's slow to get results. There may be insufficient information for making a prognosis. Genetics isn't everything. Environment's important, and other genes may modify. And probably most frustrating for both us as physicians and even for you as families is that the translation into therapies is slow. It's a step to finding out what the cause is, what's going wrong, and how we can change things, but that's not always an instant, oh, here's what we need to do to fix it. But it's a first step in that direction. So now there are some newer methods that are coming on that you may have heard about, and Aaron's going to talk about in a little more detail. One is called whole exome sequencing, and one's called whole genome sequencing. These are very powerful methods where we can take somebody's DNA and run it through a machine and get a printout, essentially, of all the changes that are there. Anybody ever see the movie Gattaca? Okay, so there's a wonderful scene in there where Ethan Hawke's character is, Uma Thurman is sort of interested in him. She manages to get one of his hairs and takes it to a little kiosk where she hands it to the clerk, who runs it through a machine and comes back with a printout this thick of what his genetic potential is. And she says something like, oh, you've got a good one. So we're getting scarily close to that. The problem is we really don't know how to interpret the information and say you got a good one or you got a bad one. So, but this is sort of the technology. It's a very cost-effective means for characterizing all the genetic variants in an individual. This might let us get to answers to genes and variants of interest faster, but it also can be too much information because you find things that you don't anticipate, and it's very difficult to necessarily sort out. So with that, I'm going to let Aaron take over and go through an example of this so you can sort of see how this works.
Thank you, Larry, and thanks for inviting us to speak. And we're going to leave quite a bit of time open for discussion because we want to get into a little bit of discussion from the families about gene testing and gene sequencing. So as Larry mentioned, exome sequencing is really now part of the common vocabulary, not only medically but also in the non-medical community. And there are lots of companies that are offering exome sequencing. You just send us a sample, we'll send you your sequence, and they'll do it for relatively reasonable price. So as Larry mentioned, it's the exome is the entire protein coding sequence of the genome. This is a misprint. It's only 1% of the genome. So about 30 million nucleotides out of the 3 billion nucleotides or 3 billion letters in the human genome. So we're looking at about 1% of the genome. Now, when we're sequencing the exome and trying to analyze the data, there are lots and lots of assumptions that go into this. And so the assumptions are reasonable to start off with, but what you're trying to do is take 30 million pieces of information and condense it into something that's meaningful and interpretable, at least that we can grasp in with just our own with our brain. So I think when we're looking at a particular disease, the first assumption that's made is that we're looking at a disease that's being caused by mutations in a single gene. So that's the first assumption. And, you know, for many diseases that we know of today, that's correct. But part of that has been limited by our technology and our ability to look at these things. So we don't have good ways of trying to understand whether multiple genes are interacting right now. So we have to focus and make the assumption that the disease of interest is being caused by a defect in only one of these 20 to 30,000 genes that we're looking at. The second thing is just by nature of what exome sequencing is doing is we're looking at variants that are just in the coding regions or the parts of the genome that encode amino acids or the building blocks of proteins. So as Larry mentioned, there's lots and lots of DNA out there that we're not looking at that has very important functions, but we're only looking at 1% of the genome, and we're making the assumption that whatever's causing the disease is in one of these exomes or the coding region, the part of that's encoding a protein. Now, there are a couple of ways of trying to interpret this even further. Most of the diseases that we deal with, as Larry said, we inherit in a recessive fashion, so basically inherit one mutated copy of each gene from a parent, when put in combination, leads to disease in the child. So many of the algorithms that we use to try to filter the exome data use this assumption to understand whether or not, or to understand if a gene is causing disease. Now, there are ways of doing this that assume a dominant expression, so either a spontaneous mutation or a mutation that's been passed down through families, but you only need one copy. But then it becomes much, much more difficult, at least from a bioinformatics standpoint, because we all have single variants, as Larry said. Probably, you know, we differ from each other at about 1% of our genome. And so you're going to get lots and lots of variants, and I'll show an example. And some of them may actually be disease-causing, but we don't know it. But at least usually when we're looking at some of these diseases to begin with, we're making that assumption that it's inherited in a recessive fashion. And then the last assumption that we make is that these are going to be rare variants. So as we said, we all carry variations in our genome. Some of these are very common. So there's some variations in the genome that 49% of the population carries. There are other mutations or variants in the genome that only one individual in the entire world carries. And so because we're looking at rare diseases primarily for the purposes of this group, we're making the assumption that the mutations or the variants in the genes that are causing these problems are also going to be rare. And so you can set your filters as rare being present in less than 5% of the population or present in less than 1% of the population. And it, you know, where you set your filter will determine how many variants and how many genes you're going to be looking at. Now, there's been lots and lots of press about this, and under the best of circumstances using these assumptions or others in large families with very well-characterized phenotypes or disease expression, the likelihood of exome sequencing giving you an answer, a single answer to what the genetic basis of a disease is about 20%. So that's even with all of these assumptions in place. So that means that we have about an 80% chance that we're not going to find anything due to any number of different things. And most of it has to do with maybe we don't have a clear enough description of what the disease process is, or secondly, some of our assumptions are false or not completely correct. The typical price for an exome right now is anywhere between $1,000 and $2,000. There are some companies that are offering limited exomes, and by what I mean by limited exomes is they're only looking at genes for which there's a known function. And as Larry said, we only know about a third of the genes do. So if we have 20,000 genes, we only know what 7,000 or 8,000 of them do. That means there's 12,000 or 13,000 that we don't know about. So what the limited exomes will do is just look at the genes for which there's a known disease association. And so you're basically looking at about 7,000, 8,000 genes, and those can be offered, you might see those advertised for as little as $300 or $500. But like I said, there are companies that are offering these on the web. You just send us your DNA and we'll be happy to provide your exome for you. They don't provide you anything else, but they'll provide your DNA sequence. They'll provide risks for various things, but in general, it's not particularly helpful without a broader context. So let me just give you an example of an exome sequencing project that we've done just in the last 6 months in our own lab. This was a baby that we had back in 1995 who presented to us with severe respiratory failure. He looked very much like at the time, a severe surfactant protein B deficient baby, and in 1995, SPB deficiency was the only thing that we knew about. But he had refractory respiratory failure, so the neonatologists did everything they could to, within their means, to try to treat him, and he didn't get better. He didn't have any mutations in the surfactant protein B gene, and he was referred to our institution for lung transplant because at the time, St. Louis Children's Hospital was the only institution in the country where lung transplantation was done for infants with surfactant deficiencies. We had transplanted the first babies with SPB deficiency in the year before that. And so we were getting referrals of lots of babies with refractory lung disease who looked like SPB deficiency. Well, he didn't have SPB deficiency. He underwent lung transplant at 3 months and actually is right now our longest survivor for lung transplantation. He's 18 years old. Well, every time a new gene came along, which we thought might be able to explain his lung disease, SBC, ABCA3, thyroid transcription factor, we went back and we sequenced the genes, nothing came up. So we did an exome exome sequencing in the last 6 months. And so this is an example of what we had. So, so I'm going to walk you through this assumption and algorithm. So when the machine first spit out all the sequencing data, there were about 18,000 variants in the exome. So then we took the couple of assumptions. Number one is that the variants had to change the coding or the amino acid coding change, so the protein sequence, or there was a insertion or a deletion of one or more nucleotides in the DNA sequence. Because these are the types of things that are most likely to be disease associated. So then we further took the assumption, as I mentioned before, that these were going to be rare because there are very common variants even within these individual groups. So for just somewhat arbitrarily, more than anything else, we chose a 2% frequency. There are databases, as exome sequencing has been done, there's databases that have on the order of 6,000 to 7,000 individuals for whom we have complete exome sequencing data. We don't know anything about what their phenotype is, if they're normal. Most of these people were enrolled as parts of other large studies. The Framingham Heart Study is one of the main ones. But these are all adult studies, and so these are really adults who are in the in this sequencing database. So we compare our results against these various publicly available databases and just select out the variants that are less than 2% in this case. And we came down to 1,098. Now, sometimes when the predicted amino acid is changed, it doesn't change the function of the protein. And so there are various, again, bioinformatics algorithms that can be used to try to predict whether or not the change of a single amino acid in that protein is going to affect protein function. Now, these are strictly bioinformatics algorithms. They also make a large number sets of assumptions. Some of them are reasonable, but these are not by any means 100% predictive. And so we know, for example, that there are variants that these algorithms will predict are deleterious, have a function on the protein, but we know that they occur commonly in the general population. On the other hand, there are some of these variants that these algorithms will predict that they're benign, but yet we see them in children with lung disease all the time. And so what do we do with that information, how we reconcile that? And we generally go on the side of if the variant's been seen in association with disease, we'll accept that variant. But even so, we do that. We still have 305 variants here. And then if we then assume a recessive inheritance, so first of all, we'll have to identify genes that have more than one variant in them. So we come down to 21 genes, 45 different variants. But then we need to have the parent's DNA to help us to go to that next step. So we'll do exomes on the parents and then utilize only those variants that each parent carries one copy of in that same gene. So this is the parental transmission. And in this case, we came down with what came out at the end of that was 6 genes. And so then we get a list of genes that looks like this, combination of letters and numbers. And one thing that I've learned is that the more letters and numbers that are associated in a gene name, the less is known about it. And and this is what's typical about the list of genes that comes out with exome sequencing. We've done many others. Sometimes we have 10 genes, sometimes we have 20 genes that make it through these various selection criteria. Now, in almost all cases, these genes either don't have a known function, or if they do, it's not a function that you would seem to necessarily associate with what you think is the disease that you're interested in. So, for example, if I recall, this is a carbohydrate metabolism enzyme gene. We're looking at a baby with lung disease, and I can't immediately relate carbohydrate metabolism to lung disease, just based on what's known. Some of these have been associated with other diseases that are completely unrelated to lung disease. But this is, again, a list of what comes out. Now, in this particular case, this is an intriguing gene because it's involved in protein transport within a cell, protein or other molecule transport within a cell. But there's nothing in the literature about this gene in terms of, certainly nothing in association with lung disease, very little known about any other association with lung disease. It's a gene that was, and a protein that was originally found in yeast and is known to traffic cellular subcomponents within yeast. But it makes sense in terms of what we're thinking about in terms of surfactant metabolism, because surfactant is a lipid as well as a protein that is synthesized and has to have a specific trafficking pattern through the cell and assembly of other components and so on. So at least intuitively, this makes a little bit of sense, but we don't know just yet. So we have a potential gene in this case. Now, this, first of all, kind of this whole pipeline from the time we got the DNA to the time we had the first sequence come out was about a month to 6 weeks. And as you'll hear lots of people say, if you haven't heard it already, getting the sequence is the easy part. It's what you do with the rest of that information when it comes out, that's the hard part. So we've been working now for several months trying to figure out, okay, how can we really understand if this gene is truly the cause of this particular child's lung disease? And so we have a lot of steps that we're going through right now. But the first thing is, we have a gene, so what are we doing? So, let's look at other family members. What if other family members have exactly the same mutations in this gene, but yet they're asymptomatic? Then we think, okay, well, then this probably isn't a likely candidate. So in this particular case, we sequenced, there's a healthy sister who doesn't share the same genotype with the affected patient. Each parent, as we said before, has a single mutation. We didn't have any other extended family members. So, so we have some circumstantial evidence that there might be something here further. The next thing we do is search all the databases, try to find out what's known about the gene, as I've said, and this particular one makes some intuitive sense. Now, is there a way to test the function of this mutation? How can we test to see if this, first of all, if this gene might be related to lung disease, and secondly, if these mutations have anything to do with the lung disease? This is where we are now, and the challenges that we're running into are, number one, is that since so little is known about this gene and this protein, we really don't know what the best cell type is to look at. So there are a lot of different ways that we can test these. You can have mice that have had their genetic makeup altered, so transgenic mice. We know that if mice have had this gene knocked out completely, so they don't express it at all, they die in utero, but that's all we know. There are many different types of cell cultures that you can use. There are some lung cell cultures, there are kidney cell cultures, GI tract cell cultures, all kinds of different cell cultures. Well, we're interested in a lung type of disease, so we're going to use a lung cell culture. The problem with lung cell cultures is that we don't have the best type of cell, so the type of cell, the alveolar type 2 cell where surfactant is made, there really aren't any good cell lines right now that we can use to test these mutations. There are some type 2-like cells that we can use, so that's where we're starting. Now, the next step is then, okay, so you choose your cell type, you try to put a mutation in, you try to disrupt the function of this gene in some way. Then you want to say, okay, does disrupting this gene affect cell function? Well, in some cases, that's appropriate if you know what you're looking for. But in this particular case, we don't quite know what we're looking for yet. So we've had some initial studies where, we've knocked down these various techniques to knock down the expression of this gene in cells, and the cells have died. Now, is that because of a technical problem in our lab, or is that a real biological phenomenon? And I don't know the answer to that yet. We'd like to think it's some kind of a biological phenomenon, but more often than not, these kinds of things are just technical problems. And then what are we looking at otherwise if the cells aren't dying? What are we going to look at in the cells in terms of what the function of this is? And so we don't have a clear answer to that as yet. And so that's kind of where we stand in trying to figure out whether or not this gene is causing the cause of the lung disease in this particular patient. we are going back. We have a large panel of patients, children with lung disease that's unexplained. So Larry has a bunch of samples. We have a bunch of samples. We're now starting to sequence this particular gene in those samples. And if we start to find other patients within this next set of samples with unexplained respiratory disease who have mutations, well, that's going to increase the likelihood that, that this is still causative. It's still not going to make that ultimate link, but it's going to increase the suspicion. We have some other epidemiologic tests. We've not, to try to understand this, we'll look at some other populations as well to see if these mutations are present. But this is, like I said, this is where we are now in this particular case. And this is something that has to be gone through every time you do an exome sequence and you come up with something that you don't completely understand whether or not it's the cause of the disease. And so when you read the papers about we did exome sequencing and we found this, and you know, we performed a miracle understanding the cause of this disease, that's a very, very unusual circumstance. This is the main part where it takes a lot of time to really understand if a gene is the cause of the problem. Then finally, once we understand this, does this really make sense in the context of the symptoms that a particular individual might have? And so it could be that we're doing all of this work and we find absolutely nothing that relates this to lung disease, and then we're back to the drawing board. You know, do we go through this process for each of those other genes on the list? I don't know as yet. So I just wanted to give you a sense of the process that we have to go through, and why you may not get an answer all the time when it comes to exome sequencing. Like I said, it's something that we all throw around and like, okay, well, this is just standard of care. We'll get the exome sequencing and we'll know everything. No, it's not quite that simple. And then the next big thing, you know, assuming we can find the gene and really understand what it's doing, then the big thing is really then translating that into therapy. And that's why you're all here, is really trying to, what is it that we can do to understand what's going on, what the underlying problem is, and then how can we fix it? And so we have to start with either cell-based studies or animal studies, trying to understand whether there are various therapeutic agents that are available. One of the holy grails right now is to really develop these large functional-based assays where you can take cells or primarily cells probably, or other chips, where you can then assay these large panels of various chemicals, compounds, drugs, and then look for some kind of a positive result that then you can then narrow down to then try to develop into a therapeutic intervention. But so you really need high, either a very specific mechanism that has a drug that's already available that you can then test for this particular indication, or use these chemical libraries, which still are a ways away. Or the chemical libraries are there, the high throughput functional assays are still a ways away. Then it's translating these into humans to make sure that they're safe, and then the larger scale trials. So, you know, much as we like to think we know a lot, and we have made tremendous progress over the last 10 years in really understanding some of the genetic basis of interstitial lung disease and diffuse lung diseases in children, you know, we've still got a ways to go. And, you know, as I've alluded to then, you know, every time we do one of these sequencing tests, it's not really just going to be a matter of sending these off to the lab, waiting for the results to come back, and then giving you the results. This is really going to require significant effort on the part of not only the clinicians who are taking care of the individuals, But the geneticists, basic scientists who really can do the functional testing to really understand if these genes are causing the problem, the bioinformatics people who have great tools available, but they're not perfect, and so there's a lot of refining of these tools that needs to still happen. And then ethicists. And this is where I wanted to open up the discussions a little bit, because as we're doing more and more exome sequencing, there's a lot of questions that come up, and these are questions that are debated in the medical literature as well as the non-medical literature. The New York Times, Time Magazine talks about some of these issues. And so, some of these considerations are, where does research end and clinical science begin, or vice versa, where does clinical care end and research begin? And in some cases with the exome sequencing, I really don't know where that is. I mean, there are places there are labs that are certified clinical labs doing exome sequencing, but how do you translate that information to an individual? You have the team, as I mentioned, but is that really research, or is that really clinical care, especially if you don't have a gene that you really know is the cause of the particular disease that you're looking at? One of the things that is also very, very apparent is that, we might be able to find variants, as I said, but unless these mutations have been seen previously in association with disease, so unless the person has the common SPB mutation, which we clearly know is a disease-causing mutation, a lot of times we're going to come up with variants that we really don't know, if they're associated with disease. And so we really have to understand that there is going to be a degree of uncertainty, and Larry has alluded to the variable penetrance. Okay, just because you have a mutation doesn't necessarily mean you're going to come down with disease, or it doesn't tell you when you're going to come down with the disease if you're going to come down with it. And so, so you can have that information, but it's not necessarily going to help us predict what's going to happen. Now, in the cases of some lung diseases, if you have an SPC mutation and don't have any symptoms, we can recommend that you don't smoke, that you don't work in a coal mine, that you try to avoid exposures, you get your flu shots and things like that, which are things that you would do under ordinary circumstances anyhow. But we aren't going to be able to say, well, be prepared to have a lung transplant in 25 years. We just aren't going to be able to tell you that. And so, so then it gets very dicey then when it comes to really trying to use this type of information for making clinical decisions. Again, unless you clearly have precedent in terms of that. And so these are some of the big questions that come up. The other question that comes up is really the genetic confidentiality. And even all the research protocols go out of their way to talk about protecting the genetic information and trying to assure everybody that nobody's going to be able to find out who you are. It's become clear that that's not the case. And there was this recent case where it was an MIT student went into multiple public databases and was able to identify through putting things together who the individuals were who participated in studies. Every study was completely de-identified in the ways that they were supposed to be, but because of one thing or another, he was able to identify those. So, we aren't as isolated as we might like to think we are. And so there's concerns about that. There are laws in place to try to protect genetic information, number one, prevent genetic discrimination, number two. But there's this degree of uncertainty and discomfort in terms of, access to genetic information. So they're not as protected as once thought. And then the last thing is, as we do these large-scale sequencing, we're going to find things that are completely unrelated to the disease that you're interested in. So if we're interested in looking at lung disease, but then yet, we have this information that you carry the genes that increase your risk for Alzheimer's disease, what do we do with that information? You didn't ask for that information. We weren't looking for it. It just was there. So what do you do with that information? So these are some of the issues that are debated very profusely and are going to continue to be debated. They're good discussions to have because we absolutely need to be thinking about these. But one of the things that that arises and has arisen in each step as new things come along through trying to protect individuals who are participating in research, the list of regulations and hoops that have to be jumped through get more and more onerous, with good intentions. To, again, try to protect individuals, number one. Number two, to really make sure that people who are participating in research studies really understand what the research means, what it will tell them, what it won't. The big thing with genetic research now, and this gets back to kind of where does clinical medicine end and research begin, is you're participating in a genetic research study. Most of the pure research studies don't allow us, our ethical boards don't allow us to return results to the families. And this is where I'd like to kind of get your opinions. So there are, they don't allow us to return results unless the results have been validated in a clinical lab. Now, some of the gene tests are not available in clinical labs, and so they're only performed on a research basis. Our ethical boards won't let us return those results to the individuals. So, there is a little bit of a disconnect, at least in my experience, and Larry, you can talk about this as well, and I know many of you are participating in research studies as well, but I find that there's a disconnect between the families I deal with and their desire to want to know what's wrong with their child on one hand, and on the ethical boards that are governed by ethical considerations as well as legal ramifications from Office of Human or federal government or other governmental agencies. And that disconnect, I think, is something that's really ripe for further discussion, especially in light of many of these ethical debates that are occurring. And so I'd like to get some discussion, some questions, something going about what your thoughts are about all of this, where we stand, especially as these large-scale gene panels are going to become part of routine care. How do we deal with these concerns?
A huge motivation for my family to participate in clinical research is that someday we could get something out of it. I don't want to say what's in it for me, but what's in it for me. So I know my feeling is really strong on this, and I'm sitting here with lumps in my throat because we would love to know. And I know that the way things are now, we can't, but sometimes I think we feel very desperate for answers because we want to provide the best we can, and we hear, I don't know. So I think it would be nice to be able to get something back. And, even if it means sign a stack of papers a mile long, okay, I would do that to get a little bit more information, even if it meant we're sorry, we can't find anything else for you. And oh, by the way, you're going to get Alzheimer's. So, because at least I could forget the heartache.
There's still a gradient there of what's allowed to be returned. And it's kind of a little right hand not talking to the left hand, because I can return the information, and it's not efficient.
Right.
Okay, so you're not allowed to do anything with it. But we can talk about it in an informed context about what it might mean. But this is all an evolving dynamic, and feel free to talk to legislators and people who run these boards in terms of what you want as parents and as advocates for your children. Questions?
Well, I have a question/comment, and I made the same question of Lisa Young earlier. So you were talking about research in the clinical medicine. So there's some of us on the clinical medicine side who took our children to a geneticist and said, hey, look at my kid's genes. So in my case, I have 2 children with very rare, kind of odd, similar symptoms. So of course we're candidates for, whole exome sequencing and stuff like that. But my question is, my geneticist is not a child doctor, and he doesn't primarily look at lung patients. So he may be finding something, but has no idea what to do with it. My pulmonologist is not a geneticist, so he may not know what to do with it. And I'm stuck with, you said, here's your child and your DNA and his DNA, but how do I get that to researchers and said, hey, here's some free data. I don't know if it's worth anything to you or not, and I know there's others in the room who've had genetic testing apart from research. So I don't know. So that's a comment.
Yeah, no, it's an important one and a dilemma for a lot of us. And part of the problem is I had on sort of my slide, one of mine, there's often too much information and sort of trying to keep up with everything. And as much as we try and keep up, I'm not an expert on everything that's going on genetically in every genetic syndrome, nor does every geneticist know the nuances of what's going on in my specialty in neonates or with lung disease. So it is important that they sort of talk to one another. But knowledge is also cumulative. So it may be that things don't make sense this week, but then there's a paper next week which links things. So it's reasonable to keep going back and just asking, is there anything new? Can you look at the data again?
So I guess
And
keep
see,
talking to doctors, because since we are not part of our research, I'm like, I don't want my data to get lost somewhere if it ever makes sense to end up in a clinical paper.
Right. But what I would also say is to urge you to keep going back to the geneticist and the lung doctor periodically and just saying, is there anything new that, looking at the data again may make more sense now? And that happens. Okay. We go back and we don't know what things meant at the time, but we get more knowledge from animal studies, from things that other people have done, and things begin to make more sense. And we're sort of all moving on, and it's helpful to us actually to say, hey, can you go back and look again?
And last comment for me is saying, how do we breach that between parents and researchers and doctors? Is I think the database for our foundation would be huge on that, saying, hey, here's me, here's my kid's whole exome sequencing if you want it.
Yeah, and I was going to say that that's exactly the conduit to do this. And I think participation in this particular foundation and this research network is going to be exactly the way to do that. All day yesterday we were talking about not only trying to figure out how we can make the databases robust, but there is so much interest in really trying to understand what these children with undefined lung diseases have. And that's going to be the basis of a lot of proposals that we have going forward to try to get funding to do. And it really is that type of thing. I mean, this case in point that I provided, this was 1995. I've not forgotten him, and I mean, we talk about this particular one every time something new comes up. We have another, I have another set of samples that was referred to me 10 years ago, we also, they stuck in our mind. We just have to find out what they have. We did exome sequencing. I think we've maybe found some candidates. So it's going to be that kind of doggedness. And the challenge is that, us as being researchers, we have a lot more time to think about these kinds of things than a clinician who's seeing 20 or 30 patients a day. And so, it's fair to go back and say, hey, anything new, or is there anybody new to talk to? But I think the main thing is through this foundation, you have access to people who are thinking about this kind of thing all the time. And even though we may not have an answer for you today, we're going to keep working for it until we find something.
And also sharing what we know is new. Okay, so things that aren't in the medical literature yet, but things that we sort of know are coming, so that we can sort of link those things.
On that sharing note, we went to see a geneticist, and basically, and I spoke to you about it actually, over the phone though. We're in Canada, and when we went in to see the genetics person, they kind of said, what are you doing here? There's no test for you. So for the people who are trying to advocate having this database with the information, what kind of thing should just us regular parents go in to these genetics people and say, this is what we need you to do? If they're not information sharing and they're not listening to all of the other genetics people out there, what can we say? Well, this is what you're going to do.
Yeah, and again, I don't think that people are trying to be sort of uncooperative, but it is to try and keep up with everything that's going on all the time is a fairly daunting task. Mm-hmm. And so the way to do that is, you can feel free to give them contact information about, here, I got this from this doctor, or my child's lung doctor, and can you talk to them about what might be reasonable to look for? Because what they'll do is they'll go and they'll do some searches from various internet databases, but even those there may be some lag, okay, between what things that are sort of preliminary and known about. And then there isn't another important part here, is that there's often a gap between what we can do research and then when it becomes clinical. So as Aaron said, what we've done comes under research studies. But if we find a new gene, even if we are 100% convinced we've got the right one, okay, there may not be a clinical lab that your geneticist can order a test for yet. Okay, we have to get that, we have to convince a clinical lab to set up that test. So that can be the barrier sometimes too. Now, we're usually successful in getting that, but it's not something that happens overnight.
I like the child database. I think it's going to be very wonderful for our families when it becomes available, and if we can submit all that information. Our children and us, we're in the Duke exome sequencing, but they're looking at ciliopaths. So as you were saying, they're going to put in certain assumptions. Mm-hmm. They're not necessarily looking for what you would put in for assumptions. Yeah. So they may not come up with anything for us. It's been a few years and we haven't come up with anything, but all three of our children have different things. And I was listening to the talk on filaments and how it affects various parts of the body, and our child, as each one progressed, each one has different varying things, but the youngest has the worst off with neurological, cardiac, right, lung, everything being affected. And I think other families have the same experience where one child may have lung disease, another child may have a neurological disorder. And when we've seen all the specialists and we talk to them about it, we feel like it's all connected. They all have some type of genetic disposition to these diseases, or they're showing up in different ways. And two of our children have 2 mutations of ABCA3 surfactant, but they're different than each other. And like you said, many of us carry those mutations, and they may not show up in myself or my husband or someone else, but they show up in the children. So, but we have no outcomes for them right now. But I think that it's helpful to have on the site and other things to have like the protocols. Like, okay, if a child shows up like this, it's good to do a brain MRI to look for other issues, do an echo to look for cardiac abnormalities and things like that.
This goes beyond even sort of the Child Foundation, but one of the things that's needed is a way to put all this information together, so that, you know, you can start to write kind of algorithms to kind of pull this out, so that we're basically putting all our heads together. It's a much more daunting task than it sounds. One of the problems that people are running into with whole genome and whole exome sequencing is where do you actually keep the data? We're not talking about small amounts of computer data. We're talking about each of these. If you do whole genome where you find 4 million variants in an individual, and each has to be annotated with the gene and what it is, you're talking about sort of a hard drive for each of those pieces of information. So how do you put all that information together? How do you link it within the clinical symptoms so that one can start to sort that out? The good news is that there are places that are starting to do that. Okay, they're trying to take all this information, putting it into databases, interfacing it with sort of clinical symptoms. And so you had yours done at Duke, somebody else has theirs done in Nashville, somebody else has this done in Baltimore, somebody else has it done in St. Louis. Can you start to pull this together and say, well, here's a common clinical theme, here's a common gene. And so right now we're all a little bit like sort of trying to describe the elephant, different parts. So
I think it's helpful too for us as parents to know with the information, because then we all communicate and we can say, oh, my child has this variant. And they'll say, oh, my child has that variant too. So it helps us to link the information. So it may not be available on a server and database in a building yet, but as parents, we can communicate through our networks online. I'm on lots of rare disease things because my son has a syndrome without a name. So I'm always looking for other children just like him, so that way we can communicate and learn from each other. And they're doing the same thing. So I think us as parents are going to be very helpful in connecting the dots for everyone.
I won't disagree.
Well, and as you're already aware, parents have a lot louder voice than we do. Whenever we try to lobby for these kinds of things, we're viewed as being self-serving. So, you know, when you have parents who are advocating for their children, that has a much, much stronger message than anything we could ever do. And I think that's, again, the power of an organization like this, the power of having Greg Porta directly in the face of congressmen on a regular basis. I think that's really going to be fantastic, and that's really how things are going to move along. So if I could just make one last plea. So one thing that came up yesterday in our discussion, and this is a task that you can all. We were talking about some of these ethical issues with genetic sequencing, and you know, some of the limitations that we have. And we were thinking that it would be nice to get a statement of some type from the parents group about, you know, what genetic sequencing means for them, what information you would like to have access to that you can't have access to today, and how the current environment is potentially limiting what you're able to have access to. And I think, you know, I'm not sure exactly where we would go with that, but you know, I think that that's something that we can discuss going forward. But I think, again, it's going to take, and I'm not trying to bash our ethical boards or anything like that. I understand both sides of the issue, but having been an advocate for children and really understanding parents wanting to desire to know and doing anything that they possibly can to know what the answer is. You know, I think those voices need to be heard, and so if that's something you all want to discuss and see about taking forward, that would be great.
Yeah, I think on that note, I mean, this is the first time I'd heard that request, but I think an easy way to start going about that is I know several people here have participated in research on the genetic level and have had, you know, I mean, we've had some things in the past, like with some people that have submitted, think that they have, like, for instance, an ABCA3 mutation that turned out to be not clinically significant because they were interpreted incorrectly by outside clinicians and things like that. I know there's people in here that have had experiences like that. I see people nodding their head. So maybe tomorrow at the round table or maybe at dinner tonight we can get a list of those people and we can collect stories and think about ways that we can compile something like that. I mean, I know that, and I know you guys know of people on Facebook too that have story after story. So if somebody could volunteer to collect those stories and we can start, I think, putting together a pretty powerful statement, because a lot of us struggle with that. So we'll get on it. Thank you, guys.